I am going to talk about how, I'm coming from the King Lab. The King Lab and the Baker Lab have historically made a lot of different types of protein nanoparticles. And so today I wanted to share a little bit of what that design pipeline looks like, and also give a little bit of context for these nanoparticles, where they, what are they, what do they look like, and we also have some nanoparticles that are not designed. And so I'm going to try to give an overarching view of protein nanoparticles, and then we'll get into the design a little bit too. So to start out, I wanted to first talk about what I mean by protein nanoparticle and self-assembling protein nanoparticle. And so I'm just showing here an example of an icosahedral nanoparticle with trimeric building blocks. So we call this an I3. It's one component. And so how we produce these when they're one component is you have a monomer, and you're just going to make that monomer over and over and over again in your cell. And that monomer has a trimer interface, and that trimer interface promotes trimeric self-assembly. And then the trimer also has a nanoparticle interface. And so that's on the outside of that trimer. You have a nanoparticle interface, and that interface is designed so that it can promote the self-assembly into an entire protein nanoparticle. And so the three steps here kind of happen naturally as you're producing this protein. If you have multi-component nanoparticles, you would produce the different pieces separately, and then you would combine them all in vitro after you've produced and purified those components. And then the last bit is, once you have a nanoparticle, if you want to actually functionalize it, add a bioactive group like an antigen or some cell targeting protein, you can then add that on top of your nanoparticle, and so you get your nanoparticle with whatever you're interested in on its surface. So the monomers generally are around 150 to 250 amino acids. So they can vary in size, though. There are smaller ones like 60, 70, but those are a little harder to find. And then the nanoparticles themselves, without any active groups on top of them, are generally between 20 and 30 nanometers in diameter. These are the designed ones. The ones I'll get into first are naturally occurring ones that can be a lot smaller. So on that note, now I'm going to talk about, I've split up this talk into 4 different little categories of nanoparticles. And so I'll walk through those first, and then I'll dive into each one. So the first is the nanoparticles that are native or natural nanoparticles. So these are found to assemble and have functions in nature. So they're already, we've not designed these just exist and are out there already. And so I'll talk about those first, and then I'm going to go into evolved nanoparticles. So there are a very small number of examples where you can actually take an existing native nanoparticle or even a design nanoparticle and evolve it. So you can force it through selection to become bigger or become functional in a way that it didn't have before. And then we have what I call these classically designed nanoparticles. So this is where the computational design comes in, and where we use more established methods, I'll say, for designing these nanoparticles. And then finally, the kind of new frontier is going to be the de novo design nanoparticles. And so these de novo nanoparticles are nanoparticles that have never existed before. They're built with backbones that have never been seen before. And so that's kind of the direction that we're going with RF diffusion and everything in the future. So to start out, I'm going to start with these native nanoparticles. And there are 2 classic nanoparticles that I'll talk about that get mentioned a lot when we talk about these native, naturally occurring nanoparticles. The first is ferritin. So ferritin is an octahedral nanoparticle. It's made up of 24 subunits of dimers, and these dimers self-assemble and make a 12 nanometer diameter cage. So this is has been used and tested. A lot of organisms have ferritin, and its main role is carrying iron in our cells. And so that's one classic nanoparticle example. The other is lumazine synthase. So this is a 60mer, it's a lot more components, and the subunits are pentamers instead of dimers. And so kind of what you can see here is these dimers that form, and that's what's self-assembling, whereas here you're going to have a pentameric subunit, and those form first, and then they form the nanoparticle. And so lumazine synthase is a little bigger, about 15 nanometers, and has a different symmetry. It's an icosahedral symmetry. And so you can have lots of different shapes and sizes when it comes to these nanoparticles, but these are the two classic native ones that we refer to a lot. And then lumazine synthase its primary role is in riboflavin or vitamin B2 synthesis. So they do serve important functions in biology. Now, I'm going to use ferritin as an example to kind of give an overview of why or what the interactions are that we want to look for when we're designing these self-assembling nanoparticles. And so it's a little hard to see, and I'll try to do my best here, but it's better when you're actually looking at it on pi-mole. So I'm showing the two examples of interchain. So this is between one dimer and the rest of the nanoparticle, and then one example of an intrachain, so within the dimer itself. So I'll point out a few areas here. If you look at the interchain interactions, they tend to be relatively polar subunits. Here and here, it's almost all polar. Here you have maybe one hydrophobic group, two-ish. But between chains, it's generally very polar. And so that's something that we've noticed in things like ferritin. And if you look at within the chain, within the dimer subunit, it's way more hydrophobic. And so you get a lot more of this hydrophobic interdigitation, this kind of very sticky interaction that's happening in that hydrophobic core. And so finding a balance between hydrophobicity and polar, electrostatic interactions is really, really important when it comes to nanoparticle design, because there are issues if you go too far one way or the other. And so what we've learned throughout a lot of the design history, the past decade or so, is that one, we tend to design very sticky, very hydrophobic nanoparticle interface interactions. There are two big problems with this. One, if it's really sticky, it's easy to envision that things are going to just come together, right? If you make a very hydrophobic interface, it's easy to bring those two together. However, it can be too sticky in that it kind of rigidly just shoves them together, and you need a little bit of pseudosymmetry, a little bit of flexibility to actually form a nanoparticle. And so if it's too sticky, you kind of lose that. And then the other thing is obviously you're going to get a lot of aggregation if you have a really hydrophobic interface. The other thing is, especially in the field of like bio-related stuff, you usually need to produce these in human cells or mammalian cells. And if you have very hydrophobic interfaces, that can lead to transmembrane regions in your proteins that prevent secretion from your cells. And so those are kind of the two big issues that we have when we create these hydrophobic interfaces. However, Rosetta historically has been very good at that, and so a lot of the older nanoparticles have these very sticky interfaces. Electrostatic interfaces are a little bit preferred and a lot harder to design. So designing a good electrostatic interface with good hydrogen bonding has been very difficult to robustly get every time. And so that's why we've relied on these hydrophobics, but with newer tools like ProteinMPNN, you can actually get to a point where we might be able to start designing more and more of these less hydrophobic interactions. So if it's really sticky, it kind of prevents, you want some solvent to be able to get in there and allow for that flexibility. If you've just got a big hydrophobic chunk, you're not allowing for that much movement. And so you can create like a really good hydrophobic interface. It's not that hard to do. You just put lots of isoleucines and valines and stick that there. But if you just do that, you're not going to get the dynamics you need to actually form a whole nanoparticle. With the native nanoparticles, we've kind of established some ideas here of what we're looking for and where we want to go. And so now I'm going to move forward outside of the native ones to the evolved ones. I'm going to focus on one example. There's not a whole lot, but I just wanted to put this out there because it does happen and it is possible, so it's an interesting thing. I'm going to focus on this paper from Donald Hilvert's lab at ETH Zurich, and they took lumazine synthase and they wanted to evolve it so that it could package its own RNA. And so what they did, or how they did this, is they created variants of lumazine synthase with error-prone PCR. They then purified anything that was secreted, and they purified the capsids, so the nanoparticles that came out of those error-prone PCRs. They then treated what was out there with the nucleases to try to identify which ones were successfully and robustly packaging their RNA versus the ones that were kind of loose and poorly behaved. And then finally, they selected that, and they repeated this process over and over again to get their final design. And so they're selecting for nanoparticles that can package their mRNA, their own DNA, and then do it safely so that it's protected from external nucleases. And so this is actually really cool because this is the original, this is a to-scale image here. So the original lumazine synthase is here, and when they've evolved their nanoparticle, they got something that goes from 60 subunits to 240 subunits at the end there, and is way, way bigger. It's a simplified, I'm showing a simplified version. There's like different iterations throughout there, but it's a very cool, actually what I just showed here is their final version just to get to that last nanoparticle. So there's several different steps they did, all in the same idea of directed evolution. But for the most part, I just wanted to show that this is possible and you can get really cool designs from this. There are examples of computationally designed nanoparticles. There's one example I know of right now that they did a very similar thing where that computationally designed nanoparticle could also package RNA. It didn't actually get bigger, but this is just something that's out there and useful for anyone who's maybe interested in a less computationally heavy approach to doing things like this. A lot of what I do is to use it as a vaccine scaffold, so to use it as a nanoparticle and then put an antigen on its surface, and that has been shown to have to help with immune trafficking in the immune system because it's bigger. So compared to a soluble antigen, for example, you're going to get a more robust, a longer immune response, and then the avidity interactions that happen when you have multiple antigens displayed on the surface versus just one. So that's one example. Another thing people are looking at is drug delivery. So if you wanted to package something inside of a nanoparticle and deliver it, you can, in theory, do that as well. We've designed pH-sensitive nanoparticles that fall apart in the endosome as the pH changes. So there are definitely a lot of potential applications, but you're right, it's very hard to design these robustly, and then to actually get them to the cells they need to go to and stuff like that is where the field is going, but we're not quite there yet. So I mean, the biggest one that we struggle with is probably stability and long-term stability, making sure your nanoparticle doesn't fall apart after a day or two. I think we've seen that very stable nanoparticles behave very well, but we don't have a lot of very stable nanoparticles. Other things are like distance from the nanoparticle to the antigen. So how far spread out is your antigen compared to the nanoparticle? Those are, and then how spread out are your antigens from each other. So there are a lot of different factors that you can play with to try to optimize a given problem you're trying to solve. But yeah, there's a lot out there that we're trying to optimize. It's hard to get when you just produce one out of E. coli. If you're going to get a raw SEC curve from that, you'll see there's usually a couple other populations. There's typically an aggregation peak, and there's typically a component peak, so some of your components are not actually going to form the nanoparticle. And so there is a little bit of off-target assemblies. There are also nanoparticles that will form half, or they're missing pieces from the nanoparticle. And so there's definitely variation, but the most robust ones tend to have very minimal aggregation and component peaks, and then they have mostly your nanoparticle peak. So we can design them pretty monodisperse, but it does happen where there are other pieces to that. I guess one other thing I'll say is, especially when it's multiple component, making sure like you're combining the components together at the correct ratios, that can also really impact your diversity, your heterogeneity. If you add way too much of one versus the other, you can totally throw everything off balance. Cool. Okay, so the next thing I'm going to talk about are the classically designed nanoparticles, and so this is where we actually get into the computational part of how we're designing these nanoparticles. So I'm going to show a pipeline here for the typical approach that has been done many, many times before and what seems to work relatively robustly at this point. So there's 3 steps to it. There's a Rosetta step, a ProteinMPNN step. Now it used to be Rosetta, and then another Rosetta step at the end. The first step is what we call docking. And so this is where you take your components. It can be a 1-component system, a 2-component system, a 3-component system. It doesn't matter. And you excuse me, feed it into Rosetta, and it will dock your components into a given architecture that you tell it. So if I want a tetrahedral, an octahedral, an icosahedral, you tell it the architecture, and it's going to sample that space. And so it samples the space, and it wiggles. Usually you have 2 main degrees of freedom per component. You have your radial degree of freedom, so can you move it in or out? And then you have your rotational degree of freedom, so that's how you're rotating that component. And so those are the 2 things that I call it wiggling, basically. You're just sampling that space, seeing where these pieces can fit nicely together. And so when you do that, you then take your interface, and you've decided that these are the pieces from that given architecture dock, that form that we're interested in, and we want to design that specifically to actually self-assemble. And so this is where ProteinMPNN comes in, and you can say, okay, let's see if we can actually get a good interface using ProteinMPNN. ProteinMPNN, like I said, tends to do a little bit more polar interfaces, which we've seen and seems to be better, ideally, than the hydrophobics, so that's good. And then at the end, we go through and we take those new sequences, and we usually use Rosetta to score them and get some sort of energy value from each of those. And then we use that to identify our top-ish hits, although I'll say most of the time, the best way to do it is just to look at them and decide which ones you want to move forward with. I'll start with the second question. The symmetry question is really up to you and what the problem is you're trying to solve. So, for example, when I work with a given antigen, if I'm working with influenza's hemagglutinin, it's a trimer, and so I want a trimeric building block. And usually the best, kind of one of the easier ones to do would be an icosahedral. You could also do a tetrahedral, but those tend to be too small to actually hold an entire hemagglutinin on its surface. But if you wanted to go and do the neuraminidase antigen instead on influenza, you would have to do, that's a tetrameric antigen, and so that wouldn't work with that building block and that architecture, and so you'd have to change that depending on your problem. And so it's very much problem specific, and you can pick 1 component versus 2 components. That's a whole other question, like how do you know if it's supposed to be, do I want a singular component system or do I want one system that's multiple components? Singular component systems, for example, are better right now for mRNA, because if we want to give an mRNA vaccine and it's multiple components, it's a lot harder to figure out exactly how to do that. But if it's just 1 transcript, 1 piece of DNA or RNA, you don't have to worry about that as much. Whereas if we're producing protein for a vaccine that's a protein vaccine, and you're administering it as a protein vaccine, and it's 1 component, it can be actually harder to filter and get to the FDA standards you want. And so sometimes maybe it's better to have multiple components, filter, get it down to the right standards, and then combine them after that. And so there are a lot of pieces to this that come into play, but those are just some examples. And so now to your first question, you may have noticed that we're already starting with the 2 pieces. So like, where did we get the initial components that we actually feed through this system. And so there are three kind of main answers to this question. The historical answer has been the protein data bank. So we've gone through historically to make these cages and just searched the protein data bank. We've looked for proteins that have the right kind of architecture, which is like we want a certain kind of helical, a certain percent of helix. You know, we want a certain resolution of structure. We want a certain organism, like we prefer ones that maybe come from bacteria and archaea over mammalian. It just depends on the problem, but you can actually go through and see which structures may map to a good subunit, and then we take those and we feed it through that pipeline. So that was the way we used to do it, because we didn't have structure prediction tools and we didn't have ways to actually make a structure without actually having a PDB crystal or cryostructure. So now what it's changed to is using predictions. So if you can maybe use homology, for example, as a way to identify sequences that might map to good components, you can use AlphaFold then to predict the structures you want, and then use those predictions to make your nanoparticles. And so that is something that we've recently done and should be published soon. And then the most current future direction is, well, we don't actually need anything from nature. We can just design components de novo and make these symmetric oligomers that Amrita just had referred to. We can just do that and make them and move forward with that from there. So before AlphaFold, we just used the oligomers that we found in the PDB. So there was no homology. So if you were to do a homology search like the second one here, yeah, that would also be those native structures. The structures that you find that you think are good structures to go into docking, you use those sequences as your homology. You know, enough people have designed nanoparticles where if you look at all the nanoparticles that are out there and you can kind of start to see patterns in what those substructures look like, and then you can go from there and pick that way. It depends on the algorithm you use. The newest algorithm is RPXdoc, and that is something that doesn't use, it's like sequence agnostic, so it doesn't like worry about the sequence. It does take into account the residues like like in the interface, like it looks at it in that moment, but it's actually not taking into account the whole sequence of your protein at all. So it's mostly backbone for the newest methods. It's a lot harder to get a homogeneous mixture of your given component when it's a cylinder or a sheet, because no one's really quite yet figured out how to cap that. So say I want a cylinder that's, 50 nanometers long and has a diameter of, 10 nanometers. How do you figure out how to get just that? Or how do you get a sheet that's like 10 by 10 nanometers? It doesn't, there are people working on trying to cap that and figure out ways to do that, but it's definitely a little more challenging, I would say, to get a homogeneous mixture. Yeah, so this is where these pieces come from. We're moving towards de novo. The future is de novo. So that's kind of the direction that this is going in. And then, like I said, there's different docking algorithms you can use. The most common one now, so it used to be TC dock. That was kind of the traditional Rosetta docking method. There's some others, like worms is another one. And then now the most common one that at least people at the Institute in Neil's lab and David's lab uses RPXdoc. And so that's how we make our nanoparticles. And so over the years, this has been used many, many, many times, this approach to make tons of nanoparticles of varying symmetries, of varying component numbers. So like I301 is a one-component nanoparticle that's been tested as an mRNA vaccine. The I'll talk about I5350 in a second. This here is a two-component antibody nanoparticle, so they're made up of antibodies, or Fabs, I should say. And then there are just a lot of different, I53DN5 is in clinical trials for flu. So there are a lot of these nanoparticles out there that have made it quite far. But the farthest one, the one that's made it the farthest, I should say, is this I5350 nanoparticle. So a few years ago, that was translated into a COVID vaccine, COVID-19 vaccine, and was clinically approved for humans in South Korea. And so this is the first case of a vaccine made from a computationally designed nanoparticle that is actually in humans today. And so this was a very great step forward because it shows that this platform does have promise, as long as, at least in terms of vaccination. And so this is why there's a lot of momentum in Neil's lab, at least, to try to build more of these and make them more specific for a given antigen. Great. And so the last step now is this de novo step. And so I know I talked about previously how you can use RPXdoc with de novo oligomers to do the same pipeline, essentially. But what if we were able to do this in kind of a bigger one-step fashion? And so you're familiar with this figure from our Rosetta fold diffusion paper. And the real question is, can you take this and can you make it not into a component, not into a symmetric like oligomer that's like a trimer or a tetramer, but can you actually make it into an entire nanoparticle at once? And so in the diffusion paper, they were able to do this with a 15 nanometer icosahedral nanoparticle. And so this was the first kind of proof of concept we needed to show that, hey, it's possible to actually give diffusion an architecture and say, hey, I want a nanoparticle. Can we actually just make it and skip all of the steps we talked about before? Now, the next step is this version of Rosetta fold we call Rosetta fold, RF diffusion. It's symmetric diffusion, we call it. And it's a version of Rosetta fold diffusion that basically breaks down your nanoparticle into a few key pieces. And then it takes that as, because if you're going to diffuse an entire massive nanoparticle, it takes way too much memory and GPU power to do that. And so can you optimize what you're selecting for and just take a piece of that and use diffusion to make that piece, and that would propagate into your entire nanoparticle. Now, that's still in the works and should be out soon, I think, that version of Rosetta fold diffusion. And I think that shows a lot more promise for this next generation of diffused nanoparticles where you're making things in one step. And so it's really important to figure out how you can, when you're defining your problems with diffusion, it's great and ideal to give it an architecture and say, I just want this. But if it's a huge protein or many, many proteins, it's going to take forever. And so trying to figure out ways to condense on your compute time and make it shorter and more efficient is always the goal, and make screening a lot easier. And so even though we're not at a point where this is like happening easily yet, we're still optimizing and working on this a lot. This is the direction that I think ideally we would go in and see if you can actually make these nanoparticles. Because if I wanted to make a nanoparticle for a given antigen or a given target, the goal would be to give, for example, if you wanted to have an antigen on this cage, could you take that antigen and say, make me a nanoparticle for this antigen? And so the dream would be eventually you get to a point where you can actually create that nanoparticle with the context of a given target that you're interested in. So that's kind of where I'll leave everyone. On this slide, you can see the journey we've been through in terms of history and the last, 20, 30 minutes. But I'll take any questions and can talk to people after too. Yes, so the question was, what has been studied in terms of the anti-nanoparticle response? So if you're going to give it as an immunogen, naturally you're going to have some sort of epitope on your nanoparticle that might elicit an immune response. And so how have we studied that? And we have looked at this, and there is definitely an immune response that's targeted towards the nanoparticle. But it depends kind of on the antigen, one. If you have a really immunogenic antigen, that kind of just shields and dominates the immune response. Whereas if you have an antigen that might not be as immunodominant, you might start to see a little bit of I wouldn't say an equal distribution of responses, but you might start to see an increase in response from your cage that draws away from some of these other less immunodominant responses or antigens. And so there is a play that needs to be optimized between your nanoparticle and your antigen, and then figuring out, can you shield? We've also tried shielding your antigen with glycans, adding different groups on top of your antigen or nanoparticle to try to hide it from your immune system. So there are ways around this that you can work on to get less of an immune response against your bare nanoparticle and focus everything onto your immunogen. Yeah, so the docking protocols, the newest one is RPX-Dock, and that's kind of why we've moved there. And RPX-Dock is very fast and very efficient, and so that's kind of why it lends itself to high throughput computation and really where a lot of the focus is. SIMDOF mover is still something a lot of people use for scoring and stuff like that to try to get some idea of like, can we wiggle things a little more and fix things a little more? So there is some balance that, some people are still using certain pieces of these older tech, but for the most part, it's just like it's the newest, fastest thing that works well. So we have used a different nanoparticle with a similar receptor binding domain antigen from SARS-CoV-2 that's all one component. And so there is some literature now out there that's shown that we can do this with a one component mRNA delivered vaccine. Right. So I know for the vaccine perspective, we know that the trafficking happens to the lymph nodes. And that in terms of like targeting specific tissues, our lab hasn't really focused on that too much. So we haven't done too much work on trying to target a given tissue and seeing if we haven't done the biodistribution. That's something we're definitely interested in to see like where are the nanoparticles going. But we haven't done a full biodistribution study. Because some of our architectures, like this I5350, there's a pentamer and a trimer, and so in theory you could have a pentameric antigen on that component and a trimeric antigen on the other. But making sure there's enough room and things are big enough and it doesn't cause chaos is a different question. I think, like in principle, it's not, but practically we don't have the tech we need yet to know how to launch two components as an mRNA vaccine. That's the biggest kind of hurdle because one, we won't know if does that cell, if a cell's getting an mRNA, an LNP, like how are you gonna package it? Are you gonna have both pieces of mRNA in the same LNP? Or are you gonna have two separate LNPs with two pieces of mRNA? And then, like I had said, in terms of like mixing, the ratios are really important, and sometimes the expression of the two can be off. If they're not expressed at the same level, you might get way more production of one than the other, and then you won't actually get a full nanoparticle. And so there are a lot of questions in the mRNA space of like, how do you get two different pieces to come together in the ratios you want inside the same cell? So that's the big reason why we haven't focused too much on multi-component mRNA. Yeah. Right. So you could do that, and then the question there is, do you get the same levels of secretion for each? Because now, which one is going to be second, and how does that hinder? Like, if you need a 1-to-1 ratio, for example, or else the cage won't form. Like, that's something that's really hard to. I mean, there are people out there who can test these things, but we're not like the experts there to look into that. So we've kind of stayed with the straightforward path of one component for now. The secretion problem is just, there's a lot we don't really understand about like signal peptides and how all of that plays into, there's just a lot of factors. But yes, that's true. Like, there are a lot of things we can play with.